to look at overview of the structure of cell membranes, their functions, the components that go into them, things like that. And then we're going to look at transport of things across cell membranes. Cell membranes have to get things in and out across the cell membrane. And then we're going to look at, which is closely associated with membranes, pathways for cell signaling. In other words, how does the cell know what's going on in the outside world, and how does it respond to that? That's this whole thing about signaling pathways, which are critically important in development, tissue repair and maintenance, cancer biology, and all kinds of things. A lot of cancers, it turned out, are at least partially due to signaling pathways going very, very wrong. Okay, so cell membranes. Every cell has this surrounding cell membrane made of lipids and proteins. The kinds of lipids and proteins that go in depend largely on the types of organisms. Plants have a different lipid composition than animal cell membranes. Fungi are a little bit more like animal cell membranes. Bacterial lipids are different than most of the other things. Archaean lipids are significantly different from what we find in, say, plants or animals. So we have a wide range of different kinds of membrane lipids, and of course different membrane proteins as well. Now, cell membranes are going to be very critical because several different things. For a cell to function, you have to have thousands and thousands of different kinds of molecules that are together, react with each other, and things like that. Everything from proteins, nucleic acids, ions, small molecules of various kinds, whatever, ATP, whatever the case is. You have lots and lots and lots of stuff inside cell. You have to keep that stuff together in high concentration so all these reactions can occur in an appreciable rate. So, if I take a cell here, there's all kinds of goodies in the cell, all kinds of molecules that are going to be reacting with each other to do the function that we collectively call life. Now, on the other hand, if I take that cell and get rid of the membrane and all the goodies float out into the test tube, the same molecules are there. But that test tube is not a living organism or a living cell. The concentration of all the goodies are too low for these kinds of reactions to occur at an appreciable rate. So basically, you get rid of the cell membrane, that's it for the cell. It's history. All the goodies leave out. Okay, so that's one thing. Keep everything at high concentration. Now, second thing about cell membranes. Cell membranes have to let certain things in and out. You have to get waste products out, you have to get nutrients in, you have to get gases in, you have to get other kinds of things in. So cell membranes have to be what we call selectively permeable. In other words, you let certain things in and certain things can't cross the membrane. Okay, it's like you go in some places, Atlanta's got quite a number of them in the suburban areas, those so-called gated communities. You know, a whole little town or whatever is surrounded by a wall, and there's a gate and a guard up there, and he only lets certain people in, the right people. Lots of this. Okay, certain ethnic backgrounds most likely. And you and I aren't going to be able to get across. No, not unless we know somebody up there. They only let the right people in. Okay, and keep all us plebeian riffraff out. Okay, cell membranes are like that. Only certain molecules can cross, whether it's going out to in or in to out. Okay, so those are important functions of membrane. The formation of cell membranes was a critical step in going from a bunch of organic chemistry into something you could call some type of life. 3.8 to perhaps even 4 billion years ago. We don't see this in the fossil record, but somehow getting molecules together packing them in and surrounding them by this partial barrier of a cell membrane was a critical step in going from non-life to life. Even though it's not in the fossil record, we don't have much rocks that age anyway, but that would have been a major breakthrough. Transition from non-living to living. Okay, well, what are membranes made of? Well, basically, the foundation structure of a cell membrane is a double layer of lipids with one layer upside down or inverted relative to the other. That's what we call a lipid bilayer. I'm going to draw that. We've done this before. I'm going to draw it again in just a moment. And then cell membranes have proteins embedded in it, spanning completely through the membrane, and so on. Because many of the membrane functions are carried out by these proteins. So in effect, the foundation structure of a cell membrane is this double layer of lipid molecules. And then the proteins do a lot of the membrane function. Okay, now, much of the membrane function is carried out by the protein. Now, the lipids in the cell membrane have to be, once again, in a semi-liquid or fluid state. Maybe not quite like corn oil, but more like corn oil slush. Now, if a membrane freezes up, the lipids solidify, nothing can move around inside the membrane, membrane function grinds to a halt. So membranes have to be in a fluid state. And as we described before, there are many ways of making membranes more or less fluid. You can alter the composition of lipids. If you throw in long-chain fatty acids, the membrane is going to be less fluid. It will have a higher melting point. They'll stick together and more likely to freeze at a given temperature. If you have lots of unsaturated fatty acids in a membrane, those push each other kind of apart. They can't pack together tightly. That makes a membrane more fluid. Cholesterol and plant sterols tend to make membranes more fluid. That's why about 10% of our membranes have cholesterol. Our membrane lipids, about 10% of plant membrane lipids are these various plant sterols that carry out the same function. Okay, so we have to keep the membranes nice and fluid. Now let's take a look at the base structure of a membrane. First of all, the major kinds of lipids that go in. Once again, we've covered this before when we talked about lipids, but once again, the lipids are going to be of the type that have polar head groups and then hydrophobic fatty acid tails. So you've got a polar part of the molecule and a very hydrophobic part of the molecule. And S1s are the known ones are the phospholipids. And what you have here, you have this 3-carbon glycerol background. I'm not trying the whole molecule. Coming off of you have, these are the squiggly lines, 2 fatty acid chains. These are extremely hydrophobic. And then attached to the 3rd carbon, you've got a phosphate ion, I'm not going to draw the whole thing out, and some type of polar molecule. And you have a number of different types of polar molecules. And these fatty acids tend to be about 12 or 14 to about 24, 26 carbons long in your typical membrane lipids. Okay, so what happens here? This portion is very hydrophobic, and this portion is very polar. Now that's phospholipids. Phospholipids are the major membrane components of animal cell membranes. Now plants, on the other hand, use mostly glycolipids. They do have some phospholipids, and animals do have some glycolipids, but they're lesser components. Now glycolipids, same basic principle. We got glycerol, we've got our two fatty acid tails, which are very hydrophobic. And then we have a complex polysaccharide. I'm just making a really small one here. Of course, polysaccharides, which are chains of sugars, are very polar. So those are two examples of the major types of membrane lipids in plants and animals. There are other kinds of minor lipids that also have the same characteristics of having a very hydrophobic end and a very polar end. Okay, so major components we see here. Now let's see how they're put together. Once again, I'm going to draw these things with this little convention here. Common kind of convention. And what that is among these types of lipids, we draw a circle to represent the polar head group, whatever it may be. And then two lines representing the hydrophobic fatty acid tails. Now obviously the polar stuff wants to face a polar environment. Water is about as polar as you can get. The second thing is the hydrophobic stuff wants to stay away from water as much as possible and rather be in a hydrophobic environment. So we arrange the best way of doing this is you take the lipids, have a double layer, one layer inverted upside down relative to the other. Like so, and you picture this in 3 dimensions. Oh, not that way, upside down, that's bad. Now when we have this, the hydrophobic tails are nestled together with each other. The polar heads are facing the water on the outside. Now we can add membrane proteins in here. And continue on. So that's a little small region of a membrane. Now, the membrane. Okay, now, once again, those membrane lipids will spontaneously form these lipid bilayers in like a little spherical shell because that's the best way to keep the polar stuff facing water and the hydrophobic stuff snuggled together. That's really the best way of doing it, minimizing hydrophobic to polar contact and keeping polar to polar contact at maximum. You take these purified lipids, break them up with ultrasound water, they spontaneously form these structures. Now all you need to do is add proteins and you got yourself an artificial cell membrane. Okay, so you don't have to assemble them. They will assemble themselves simply because that's the best way of keeping polar stuff in water and hydrophobic stuff out of water.